chemistry realm, there are emerging foundation models for chemical structure, bonding, conformations, and energetics. And those kind of make sense, and they are contributing, as agents to enable agents for small molecule drug design, for example. So what you've been saying leads me to wonder, what are some foundation models in biology? And they would appear to be very much more complex, but maybe there are many, many types of them that can be produced that will enable certain tasks, again, along this trajectory that we've been describing. Foundational model for interactomes. Interactomes perturb in different pathological states. Those accrue. And then that would lead us to either synthesize or collect certain experimental data to support those things.
Yes, I think that now is the rise of, in this area, is really emerging the role of synthetic data. Because for biology, we are not there, but for chemistry, we are there. So quantum mechanics, density functional Taylor simulation are really able to address the complexity of chemistry. So now the raising of the generation of this kind of data scale is changing the property, the functionals, the force fields that are behind all the physics-based simulation. So the level and the quality of the model, of the functionals trained on synthetic data is much, much higher. So now you can do molecular dynamics simulation with quantum mechanical quality because you are using neural network potential trained on quantum mechanical data. So in this part, we are there. For the chemistry and the generative chemistry and the property prediction, I think that is really a low-hanging fruit. In a couple of years, we'll be able really to model and understand the chemistry at the level that is required really to make the difference.
Yeah, I think here too, I wonder, we can simulate a single protein pretty well by itself. And if a disease has multiple proteins, how do we do this simultaneous simulation of them working in concert or failing in concert, and how this pathway, as opposed to a single target, can be simulated and exploited?
I'm not an expert in this kind of a system. I think it is a multiscale system. So there is a way when the evidence that you have at the molecular level, and so the prediction of the activity of the effect is a parameter in a multi-step models. So you change the scale in the process. So you use molecular simulations to predict the actual interaction that then is a parameter of a multi-scale model.
We can do that, and then the downstream consequence of that molecular interaction produces some effect. And then the combination of that with another downstream effect is, I think, where we're trying to get in terms of simulating a disease that moves through multiple pathways.
But the change in the mentality is that in the past we are trying to understand and have an explicit model. And then come up in the elimination our understanding of what is the system. What they are trying to do now is the reverse. So as I said, you knock out any gene in a cell line, and then you test 1,000 molecules to see the effect. So the system have the signal and the effect. And then is the system itself, so the self-learning part of the model, that they have to find a way. That the question is wrong or is good, but if it's able to predict the behavior, then you have not to care too much on what is happening in the machine.
It's not correct, but is it useful?
Yes.
Right.
My understanding of statistics is certainly it's more powerful. It's easier to be predictive than explanatory. And we will get to the predictive powers first, and that will enable many, many things that we need to do. And we had an earlier exchange kind of around, emerging collaborations, consortia, sharing data, primarily around small molecules and small molecule drug discovery programs. kind of driving a change in the notion of what constitutes pre-competitive data and information versus really competitive information. And we see in the small molecule area that kind of coming. It's much harder, I think, in biology and around targets is how do we get the community maybe to work together to kind of shift the goalposts for targets and things like that so that the whole community would enable the kinds of understandings and data that can be shared or utilized by different organizations in different ways for their own goals, and realizing that everyone wins by that kind of structure.
Yes, I think that we can have a sweet spot here if we focus on toxicity or ADME prediction, in the way that we design the molecules that give us the best models. So this is a perfect match for me for a consortium.
Okay.
Because in that case, you design the library and the chemistry that you want to expose on targets that there is no IP attached because they are anti-targets, or tox target, or alert target. So in that case, I think that we can take the maximum, because if we design safer molecules, it's pre-competitive by definition, and there is not the problem of reverse engineering or understanding what you're doing. Again, I think that if we use our own molecule, there is always the problem that I'm exposing strategies, knowledge, some sort of IP. But again, if I think it's better for the model, maybe it's better to design the library new, that have the only scope of training super efficient models. And then we have no problem. And the idea is having a consortium, because then when you have a very balanced chemical space, that you can profile and generate multitask models and do wherever you like. And there is no problem for joining a consortia where I have no IP at my risk. Now, and I take the maximum, the fact that I can continue generating data on the same data set. And so every time I add value for the new information and target, but also, as we know from multimodal modeling, that also the other part of the prediction can benefit from adding additional layer of information. So I think this is one of the driver of things. So we have to take out of the idea of reusing things from the past because it's there. So try to think in a completely different way. Say, what we need to do to make it as best as possible.
I'm thinking in the public domain are, kind of government-funded basic research efforts in, pooled CRISPR screening for, phenotypes, RNA-seq readouts that come with whole genome knockouts, whole genome activation of genes, or the really interesting emerging things around the blood proteome as a function of age, as a function of organ age, as a function of disease states. And so those are in the public domain, right? So they are there, and I'm sure companies will be leveraging those and multimodally leveraging them to bring new insights. Is there a level above those that might come from consortia that is another level that might still be considered pre-competitive? It's hard, I think, to think that for me, to think that far ahead. But that, of course, is just one more step towards kind of in this trajectory of where we think we can go. Relating to synthetic data in biology, in physiology or pharmacology, other than synthetic data around small molecule structures and properties. It's more expensive, still to produce, right? But it's more expensive to produce experimentally.
The validation, I think, is the big issue. We have our attempt to try to do synthetic data in biology, the prediction of protein functions. Because there is a lot of annotation of protein function, but it's still based on a concept that is 30 years old. So you start from the motif. The motif is a linear sequence of letters that are the amino acid. If you change any amino acid, then you can be found in any motif finder. But the structure, the secondary, the solvent accessibility, now the physicochemical property of the surface are maintained. Now, for example, with this tool, we are able to identify that the spike protein of SARS-CoV-2 is mimicking the cofactor of estrogen receptor because the shape, the property, and the solvent accessibility are the same. And we demonstrate in a paper that is related to the side effect. And now we move this technology able to identify more than 5,000 motifs. And what you see that you light up the protein with new functions. And this is the reason why we are investing so much in knowledge graph. So knowledge graph allow us to say, what if this new feature is there? Means that it pop up in another pathway that never reported before. And so we say, this is a new target. Now, how we can validate this new target in this setup? And then we come in the more expensive and time-consuming target validation. But I think that can be done. This case is not very costly because the tool is very simple in principle. We use a quite complex way to represent the surface of the protein. We have an invariant Hilbert space representation on a multistrate. So it's very interesting, but is much more effective than a neural network and have only 17 parameters. So it can be trained super easily because every layer is a property of the surface, acceptor, donor, hydrophobic, and whatsoever. So this is a demonstration where you don't need a huge amount of data, but then you can generate synthetic data that can be useful for biology.
Right, and then it provides basically an agent that is easily probed. Yes. Scientists can pose hypotheses to it and rapidly get them answered. Very interesting.